Dear Chairman, dear Professor Liskoy, I would like to say thank for invitation me to this Congress, which was really very interesting for me today till that time. And it's already a question, you know, we have heard many good lectures regarding to the new therapies in melanoma malignum. The question is how is coming with vitamin C to these new drugs, you know. And I guess that after my lecture you will understand that vitamin C is not an old thing, it is old, but for the future we are not allowed to forget it. How old is it? So the first publication was in 76, so already 15 years ago, when Pauling and Cameron published that prolongation of survival times in terminal human cancer is observed after supplemental ascorbat therapy. It was a very interesting result at that time, but after 4 years, another publication appeared. It was written by Mortel and Cregan, and this Mortel repeated this once more later after some years because they said that failure of high-dose vitamin C with advanced cancer. So he said it is not true what Cameron and Pauling said. And it was a strong debate between the two groups, of course, but Pauling was and is a Nobel Prize holder, so he was a strong person, and he was quarreling with Mortel, who said, no, you have no right, and they said that you are belonging to the crazy Nobel Prize winners and not for the real ones. And he repeated his lecture, his study, and at that time he said that he has a randomized double-blind comparison, while the study by Pauling was a match-controlled study. So therefore he said that that's a real knowledge, while we can forget what Pauling was saying. After some years, it appeared that it is not true because in the reality, Pauling had the right. And it was because it turned out that vitamin C do kills cancer cells in vitro. And it was also proved that it helps the survival of tumor-bearing mouse and animals. And it is always so, but in human beings it is not always in that way. So it is still a question. What is the reason that vitamin C always active and good has a killing effect against cancer cells in vitro and in animal experiments, while in human beings the result is ambiguous, contradictory. I suggested 20 publications, and I collected the data of these 20 publications, and I published it in 2022. And from that it is possible to realize that 9 of the papers said that the result, the clinical regression was remarkable, while 9 other papers said no clinical results. So the publication said sometimes it is working, sometimes not. So the question was, why is it? So how is it possible that animals are always doing the way how it should be, and why is it that it is not the case in human beings? I tried to answer this question during my lecture, because after realizing this question I started to think about this and finally I was writing a hypothesis which is about energy transformation. I suppose that the energy transformation in the cells are also very important, And as cars are using diesel or petrol, benzene, and not air oil, so we must transform the air oil to petrol and diesel fluid, and afterwards we can use it in the car. I think it is very similar in the case of glucose. The glucose is similar to the air oil, but the cells are using ATP instead of the glucose. Although from the glucose the ATP when is made, the energy is already made, but this ATP is the real energy source of the cell, because the cell can use it without any danger. If the glucose is transformed to ATP, the danger is there, and the cell can be easily die. So I think that in an eukaryotic cell, two types of structure for energy transformation are detectable. One is in the mitochondrium. I say it is working with oxidative phosphorylation, while the other is using aerobe glycolysis, and it is in the peroxisomes. I guess that people today forget this peroxisome working CEEAO because the mitochondrial energy transformation is much stronger and much more important. But I think that as the genetic background of mitochondrium, we know that it exists in that way, the old cell must have another structure for energy production, which is much less effective, but it is very important for us, because if we cut our tissue and the oxygen is not arriving to the cells, then the mitochondrial structure is not able to work, and instead of the mitochondria, the old one will work, and it will help us to create the new vessels. Now vascularization will occur, and then the vascularization will be there, and then the whole thing can return to the original situation. Another very important thing with the mitochondrial background is that the mitochondrium is not only producing much more energy than the old structure did, but this is also producing material against free radicals. So it makes a defense against free radicals, which is given over for the eukaryotic cells. In tumor cells, the mitochondria are not working, and if they are not working, then they are not producing the defense against the free radicals, and therefore the cancer cell will be sensitive for X-ray radiation, but only if the mitochondrium is not working. Those tumor cells which do have mitochondria in work, they are not sensitive against the radiation, and they can survive, and we can detect them after some years in the same place where the person was irradiated previously. Let's speak about these structures which are responsible for the adenosine triphosphate production. So we have to go back to the publication of Austin. He was showing that in the mitochondria there are 7 iron sulfur clusters with 4 iron, 4 sulfur. These are from this, we have 7 in this exception. In my opinion, instead of that, we have only 1 of this kind of structure and 4 iron 7 sulfur cluster is responsible for the transformation of sugar to ATP. These clusters are bounded together with special things. there are the so-called cysteine parts of the effect of these clusters, and they bound together these 10 clusters, and they have also places where other molecules can be attached. So acetic acid and glucose can be attached to that, and adenine and adenosine triphosphate can be bound also to this part of the cysteines. Here I show that 10 of these clusters are bounded together, and they will create one electron transfer chain, that is electron transfer chain 1, which will be bound to another one, to the electron transfer chain 2, and one of them is in an active stage and the other is in the inactive stage. And I suppose that after a while the active stage, when it finishes its work, it will activate the inactive part, and afterwards the inactive part will activate the other part. So in that way we have a circle which is going on continuously from the beginning of our life till our death. So we have a circle which is running always, and this is influenced by vitamin C. The vitamin C can influence the speed of this circle, and during this change, as I told, the result will be that one of the chain activates the other, which will be activated by the other, and it is important that at the same time glucose molecules are entering the structure and ATP molecules are leaving the structure. I think that the old structure is just as I showed you before, and the new one, which is in the mitochondrium, it has a pyruvate dehydrogenase enzyme, which is responsible for the burning of pyruvate in the mitochondrium, and therefore it creates much more energy than the previous ones did. It is important that the iron has two possibilities. One is when the iron has 23 electrons, and the other possibility is that it has 24 electrons. And it is said by this person that vitamin C is able to change the iron nature in the way that at the time when from ascorbic acid the hydroascorbic acid will be made, at the same time from the 3 plus iron, 2 plus iron will be created, plus 2H will leave the structure. So I guess it is very important in the activation of this whole process, and I guess that adenosine triphosphate has a very similar possibility to activate the whole process as vitamin C does. Here we show one cluster with 2 iron and 2 sulfur, and in that structure 4 H2PO4 molecules are bounded, and after the reaction this will create 2 CO2 like here, and it will create 4 PO3 molecules, which are used up for creating adenosine diphosphate and triphosphate, and it will be also some hydrogen produced, which will be responsible for the membrane potential. So actually in the old structure the structure will produce 24 CO2, in the new one 24 plus 12 coming from the pyruvate molecules will create 36 CO2. What happens if the reaction is running, but the glucose is not arriving to the structure? In that case, the oxygen, which is responsible for creating the CO2, will kill the cell. First, caspase will inhibit this killing effect, but after exhausting of this system, the cell will definitely die. As cancer cells are using about 200 times more glucose than the normal cells, therefore they are the targets of the vitamin C treatment, especially the mitotic tumor cells are the targets. So I asked what is the answer of my question. The answer is sugar consumption, because it inhibits the efficiency of the vitamin C effect. So the question comes also whether it is important for us for the future or not. You know, we have heard the new possibilities for treating cancer patients, and these are helping the patients by inducing the immune effect of the person. So if the immune cells are fighting against the tumor cells, then the result is important. How will be the result? If the tumor is creating more new cancer cells, then the number of the killed tumor cells by the immune cells, then the tumor will survive. If the immune cells are killing more cancer cells, then new cancer cells are produced by the tumor mass, then the treatment will be successful. So therefore, I guess it is very important that we have to calculate with the number of the tumor cells, because it might influence the effect of the immune treatment. If we eliminate one part of the tumor cells by treatment with vitamin C, then the efficacy of the immune system will be stronger. So therefore, I guess that in the future it is possible that we will have an infusion which will be said that okay, that works for cancer, and afterwards we should use this just as addition for the immune therapy. That was what I wanted to tell you, and I have here some information also. This is the publication about the vitamin C treatment, what I published 2 years ago or 3 years ago, and this, the energetic system of eukaryotic cells, is a brand new, which was put on the net just yesterday. Thank you very much indeed for your attention.